السلام عليكم ورحمة الله وبركاته. بسم الله والحمد لله والصلاة والسلام على رسول الله صلى الله عليه وسلم ومن والاه. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. Assalamu alaikum wa rahmatullahi wa barakatuh. Welcome, brothers and sisters. This is seminar episode, I believe, our 15th of our Golden Eight, our Golden Lab, and our 62nd, Allahumma Barakatuhu, mashallah, la quwwata illa billah, of our Golden Age. And so, inshallah ta'ala, brothers and sisters, stay up to date. We're continuing, inshallah ta'ala, bidnillahi ta'ala, an amazing program this week for BIOA02 students. They're in their third week, and inshallah ta'ala, they'll be focusing on Daphnia, which is essentially a water flea, and being able to observe physiological traits on either heart rate and how it changes with changes in temperature. So inshallah ta'ala, students will be dealing with this on compound light microscopes. It's a bit difficult to, I must say, within these labs to demonstrate them directly, but hopefully verbally, inshallah ta'ala, you'll find immense benefit and be able to interact accordingly. So inshallah ta'ala, students will start off with their pre-lab quiz, and thereafter, following attendance, they'll be given updates for their FLR, formal lab report. Thereafter, zoo field trip to Toronto Zoo, bi'idhnillahi ta'ala. And then thereafter, there'll be also thereafter they'll be essentially going over exercises from today, or actually going back. I plan, inshallah ta'ala, if Allah wills, to make reference to a few connective points to, inshallah ta'ala, inspire them to not only just think about grad school or academia and research, but importantly, through the lens of Islam, to appreciate, the divine signs that Allah has displayed for us in His creation and in these lab observations, right? So, inshallah ta'ala, they'll have a look at a heartbeat of Daphnia, and they'll actually look. Daphnia is nearly transparent, so it's very beautiful to be able to see the heart physically beating through like simple analysis, very simple, literally on a microscope, putting in a small smear of Vaseline with a glove, and then a drop of Daphnia with water. And inshallah, students will actually literally see and count actually the heartbeats, and they'll be adjusting that temperature of water over time to be able to see if the heartbeat slows down or increases over time, which is essentially a bit of their first exercise. The second, following, yeah, and then after this is their data analysis, which I'm going to talk about in terms of, biomathematics, really connecting math with biology from an Islamic perspective, bi'idhnillahi ta'ala. And students, inshallah ta'ala, will be able to interpret their data before submitting it tomorrow, inshallah, at 1 p.m. And then some closing reminders, thoughts, if there's questions as well, that they're more than happy to ask. But in the meantime, we'll pause here. Okay, so how many of you are still doing the lab quiz? Just so I can get an idea. How many more minutes do you have? Sorry, I just wanna, I forgot everyone's name. Adrian, how many more minutes? 2 minutes, okay. Anyone else on this side? How many more minutes for? Just started, okay. Uh, just started. And, so let's wait just a bit more. Um, and hopefully in about, I would say 5-ish minutes, we could get started with going through the material for today. But just bear in mind that for those that are done, make use of this time to look at the lab manual to see what you have to do for exercises 2 and 3. Essentially, you have a post lab that includes your results from today. So essentially, you'll be doing analysis of Daphnia for the physiological heart rate over different temperatures, and you'll be recording that data every 90 seconds for different temperatures from room temperature to 10 degrees all the way to 35 degrees at 5-degree increments. So you're going up by 10, by fives, from 10 to 15, 20, all the way to 35. And so essentially those results will essentially be used to calculate the mean heart rates per temperature to eventually put that in to calculate your Q10 values, which all of those details will be included, including a graph in your post-lab assignment. So just bear that in mind. And hopefully in due time, you could get yourselves ready. If you feel like you have enough time, I would recommend once your group is done, you can actually leave following the lab. But just bear in mind that we're able to go through all other parts, all slides for today. So that way you should all be able to understand what's to come for post-reading week, right? Following next week and 2 weeks after your zoo trip for lab 4. So be a big exciting for us all. I haven't been at the zoo for some time. And hopefully for those that still are doing the lab quiz, I hope I'm not interrupting you in any way, but I haven't touched on anything that won't be hopefully repeated. If not, then feel free to ask your lab members. Um, see. Okay. Are you ready to begin the lab? Um, Adrian, Ranran, are we both done? Yep. Done? And Joshua and Kiyomas, Walter, you're almost done? Yeah? Okay. I'll wait another 2-ish minutes or I'll just like go through very basic stuff so you won't miss too much. No problem. Let's go to full screen. Okay, everyone. So we'll be paying attention. Please bear in mind, no phones unless you're taking photos following my permission. Right? Like if you need a phone for taking photos or like of the slides or if you have specific questions, just let me know. But I do want to make sure you're paying attention because we only have a limited amount of time for today's lab. In fact, only 2 hours and 20 minutes. Following 11:40, we actually technically have to clean up so that way sorry, not 11:40, 12:40. Following our lab or following that period, we'll have to essentially um, clean up so that way the upcoming practical will be able to come in due time and the lab technicians could help clean up other things while you do the same. So just bear that in mind. 12:40 is our deadline. If you can finish sooner, and many people have done so, but if you can't, that's pretty much the max that you'd reach up to 11 or sorry, 12:40 p.m. Now the lab plan outline for today is shown here. So essentially, we have different things shown here, but briefly, I'll be going through. I've done the attendance. Many of you have done the pre-lab quiz. Now, with regards to your FLR, all of you have likely seen it on Quarkus, the data sheets. Use your practical 40, even section on Wednesday to be able to see your data results for the the shoot biomass and shoot lengths of the winter wheat. So remember, deadline is March 3rd, but you should actually start beginning now, at least for your results, because if I'm not mistaken, I mentioned 2 weeks ago that you can start getting ready for your intro. So now that you're done your intro, even perhaps your methods, because we already completed the practical, you can go straight away to your results section. Then during reading week, you can finish off your discussion and you're pretty much done a month or maybe at least 2 weeks in advance, right? So the sooner you're done, the better. If you have questions, you can let me know then rather than, before it's too late. So just bear that in mind. I'm more than happy to help if there's or at least guide you if there's a question that perhaps isn't answered from, um, and I'm just going to skip some slides from the lab help TA. Of course, you can ask the lab help TA. She's actually dedicated to help and serve you guys or help you guys for your FLRs and other important assignments. But just bear in mind that you can contact her even directly from the course syllabus and through the help sessions. But just bear that in mind. The other thing is, yes, March 3rd at noon. So bear that in mind. The deadline is March 3rd at noon. It's not at 11:59 p.m. So please bear this in mind because like your written assignment or writing assignment, you need to make note of the timings that they're due. Otherwise, you'd actually be penalized. And I don't want anyone getting penalized, right? Just not knowing the timing of the submission, even if you submit it on the same day. Any questions regarding that? Questions? Okay, so a bit of academic offenses or more so academic integrity. Please bear in mind, I know the life of an undergrad where it's very tense, perhaps have 5, 3, maybe 6 or 7 courses on your plate. It is important that you, especially during these assignments and down the road in your careers, you make sure you do things authentically. And that's one of the keys traits I mentioned earlier in our first practical, if I'm not mistaken, right? Authenticity is key because it delivers and showcases your true identity and how you deliver yourself and conduct yourself within the workplace or within the lab space, but also beyond into the workspace. So just bear that in mind. Indeed, the sanctions are tough. So just bear that in mind that you're not that you're being mindful of the consequences of your affairs and the consequences of your actions. for yourself and those that copy you, right? So don't use AI. Two, don't essentially replicate results with one another. Like if I find two identical, because I know and I am able to see all of your submissions, right? So bear in mind that if you're using and concocting essentially results, that is going to be on your own consequence, right? And you are literally jeopardizing your grade and also those that essentially are influenced by your mistake. So just bear that in mind. If you have questions, let me know. Once again, there are official ways of asking for help from myself, from the bio TA, Andrea. But if you want, you can ask me at any time throughout the lab or even outside if needed, ideally via email. But if possible, during our practicals, when there's open space, that will make it easier because I TA different courses and it's a bit difficult to essentially get hundreds of emails to, or at least tens of emails to respond to on a regular basis, but it's okay nonetheless. So, any other questions regarding this at least? No questions? Okay. So, um, the next, so 3 weeks from now, which I guess, yeah, pretty much 3 weeks from today, you'll have your lab 4 at Toronto Zoo. So, important note, you'll actually be going at 10 a.m., if not by 9:30, to the zoo. You won't need to, and inshallah, I hope no one comes here for lab 4. Reason being is because one, I actually mark attendance there, and two, your assignment and everything that you're supposed to do for the module is there at the zoo. So my kind request, go straight to the zoo. The address is here, 2000 Meadowvale Road. If you want to go on Google Maps or just essentially like find out even on the bus commute systems that you take, try to be there as soon as you can, 9:30 a.m. Ideally, inshallah. So that way, you get also parking passes. If you're even taking a car or carpooling with some friends, you guys can go all together. With that in mind, just bear in mind that we all go as one practical, meaning that you need to be there as soon as you can, because if I find that essentially it's past time and there's a number of you that are late and haven't actually corresponded with me directly, we'll literally have to go on and you may have to be stuck. So, or at least you'd be delayed. So I don't want anyone being stuck in the commute or in traffic. So just bear in mind that if you ideally, please come in as soon as possible. 9:30 is the date or the timing in which the zoo opens. So just bear that in mind, everyone. Um, any questions regarding Lab 4? If you also want to have a look during reading week, you can have a look, skim through of what Lab 4 discusses. It's going to be a mini scavenger hunt, perhaps, but nonetheless, have a look. Things are up to date from your manual. And hopefully, if there's questions, you can also reach out at any time. If you also have an exam or midterm during that time, let me know. Let also the biolife.utsc email be CC'd or informed as well. Now we're going straight ahead. Just want to see time. Okay. So in terms of timing, we'll just go straight ahead. Ooh, all the way down to today's practical and just go straight off to metabolic rate. Now you probably have touched upon metabolism just a bit, perhaps in module 2. But if you haven't, just pretty much to get a rundown, it focuses on biochemical reactions within a body. That doesn't just have to be in the human body, but in the case of our practical today in Daphnia, water fleas, which are actually in the middle bench. And so the idea is that, in organisms, they have different metabolic rates. Some are relatively higher than others, and they actually survive in different climates and indeed are influenced by changes in their energetic demands, right? Sometimes in aerobic metabolism or respiration versus anaerobic, which is often the case in some bacteria, but also in terms of external temperatures, which ourselves and many other mammals and even invertebrates are essentially um, uh, are surrounded by. So essentially, this graph here tries to highlight this in a nutshell, something that physiologists like Stephen Reed, Jason Brown would be able to highlight for you. That essentially that you have on your x-axis ambient temperature, external environmental temperature, and essentially on two y-axes. So bear in mind there's two axes shown here, a primary y-axis of oxygen consumption shown by black dots, and a red curve highlighting metabolic rate as a secondary y-axis. This actually can be done also on Excel, but you won't be required to, but just as a thought for you just to be aware of. The idea is that as temperature increases, oxygen consumption can increase, and similarly metabolic rate. So these are things that you want to keep in mind because it does relate to actually what we're dealing with today in the case of Daphnia. Daphnia spp, that's for the subspecies that we're dealing with today. There, as you see, are crustaceans, very small indeed, and are commonly known as water fleas. They're also transparent, so they're not in this hue, but nonetheless, importantly, they are poikilotherms. Does anyone know what that means? Any idea? It is hard to tell by actually the prefix, but essentially knowing that they're ectotherms, it's very similar in this regard in that they mimic the external temperature environment, right? Now, the thing of poikilotherms is that they are more sensitive, or at least they are more responsive to external stimuli, including at least temperature in particular here. So just bear that in mind, and indeed changes in external temperature can have changes in metabolic and biological processes, including heart rate, right? So bear that in mind because we'll be actually looking at their hearts, right? As you could see, the change in size or like palpation because it's moving, like it's opening the valves of the heart itself and then closing, compressing and expanding accordingly at different temperatures. And actually, at a single temperature, you'll see heartbeats occur. So that's something that um, I want to pause. Actually, I'll pause after. But nonetheless, something that you'll likely see in due time. Now, pretty much on your bench, this video is going to highlight what you need and what you already have. Some things are on your benches in groups of two that you'll be working with today, 2 to 3. And essentially, there are other instruments shown in the middle bench. The middle bench essentially includes the Daphnia in a small jar, which I'll be taking out for you guys. But in another jar, smaller Petri dish or glass dish, you'll be having Vaseline. So you'll be adding in a small smear of Vaseline, a thin smear. So that way we'll be adding in or placing on top your Daphnia. And I'll be helping you on that regard. But then after that, you'll be essentially using the compound light microscopes as pairs to essentially, or even as trios to, like you can do it in segregated format to essentially pursue analysis and visualization of the Daphnia and their heartbeat. So that's going to be really cool, really exciting, actually. And I hope you enjoy that exercise. But essentially, you have what you need. And then following visualization, we'll talk about what steps are next. Essentially, you'll be adjusting the temperature. But first off, let's get started. Um, I'll show as you guys get ready with the instruments that you have on your benches. I'll press on this video so you get another idea as to what's going on. Pretty much the mercury thermometers are placed on your benches as well. The toothpicks are present on the middle bench. The large and small, actually, I'm going to pause. I want to pause here. Essentially, it's actually important to note that when you're dealing with computers, essentially, they're already labeled or already organized for you in that you have essentially your mercury thermostat. So here, or this thermostat here. Make sure that you see them placed into these speakers. They're not left unattended to because if they just fall out, or just on standby on the benches, they can literally fall and break, and you don't want essentially to have anything spilling or even broken on the floor. And also because we have limited amount of materials for today and even for other practicals, just bear in mind you don't break anything. If you have a question, please let me know. But essentially, you'll be using this thermometer or thermostat to essentially see changes in temperatures, and you'll be recording with the water. So you'll be able to essentially adjust with the ice bucket and taking in small pieces to essentially lower the temperature all the way to 10 degrees. And then using the 55-degree water bath all around here, you'll essentially you'll essentially take out small amounts to adjust when you're going up. So you're going down with the ice bucket, you're going down from room temperature to 10 degrees of ice, and then and the aged water is for the room temperature, just so you're aware. But then going down with a few scoops, scapula of ice, essentially, into these larger beakers, and then essentially you'll take a few drops, just a few drops, to surround or be bended around your Daphnia, so that way you could actually see in real time the changes in heartbeat. And that's where you essentially record with these counters, and what the video will show in due time, the number of heartbeats that you see. So you'll separate, like that's essentially a bit of what today's practical is. And then just as a reminder, when you're going up in temperature every 5 degrees and you'll record that with thermostat, you'll be taking literally the larger flasks to pour in, and thereby increase temperature from 10 degrees. Any questions? Time reminder as well, make sure once again the phone, but also that your lab coat is being active. Yeah, that's just a kind reminder. Now you should be able to take those the smaller or the larger and wider or wider and smaller plates to essentially smear in Vaseline. You guys can actually get ready now. I'm just going to show the video as like a supplementary aid, but I pretty much said what you need to know. Actually, yeah. You guys in the meantime can bring your plates or these ones, these glass dishes, bring them to me with, and then you'll essentially add in some Vaseline, very small, thin smear here, and then I'll give you that. You'll do it as group, and I'll let you know. Just come in as soon as you can, and then we can get started with that. Technically, those bishop was already in and then essentially you guys will all of you just bring one person to bring in your glass dish, another person to start on the compound like microscope. Turn it on, try to get the light adjusted, and then if possible, try to get along this line so that way everyone will be able to go one by one. So the first thing I'm going to ask if you could put you can literally use the finger as well. Too much. So maybe turn a bit of it.
Oh, too much? Like that?
So can you sometimes I'm like I'm using other things because it actually is just like this is good enough. Just so you guys know, this is what a thin smear is. So that way, once I add the Daphnia, hopefully it'll stay. Just so everyone else. Now, in the meantime, other groups, actually, once you're done, you can wait here. Other groups can start adding more basketball blocks. You don't necessarily know if you want to. Well, it's reason I say you don't. I mean, you can actually. You can. The reason why it's also better actually with a glove is that way you can be more well. But the issue is you don't want to actually come out because otherwise you'd have to use the prayer. Maybe one isn't that.
Let's go.
I have no idea. It's like pretty soft.
Yeah, yeah. In the evening. Yeah. I don't believe that. I don't believe that. There's no way. I would like 10 to 25. The person to read the FS to read the person. Or maximum of the whole people. Just the next people. The person they do also afterwards. Yeah. Like they have the exam. So much trouble. Yeah, well, unless it's like, I don't know, something terrible. Right?
Can I have a look at the? This, if you want, have a look at this. I think they're good.
Go.
We can actually.
The exam.
No, I have a problem. Oh, you're trying to get that cut? Yeah, I don't know. Okay, you can come at 8:30. Yeah, today we have a picture. He's complaining about it.
You know the event, the event day, they cancel.
Yeah, because that's not necessary. Yeah, they could have postponed our exam.
Make sure that another member of your group has started to add in a little bit of the aged water into the starting beaker, the small beaker, so that way they would be able to essentially move forward with the practical. I just want to show this is actually a really good example. So essentially, you have the smear shown here, and then just 1 or 2 drops at max of Daphnia really helps. So that way you can maintain a good initial temperature, which is essentially one that you see with your aged waters. So try to have like a similar.
Oh, that one's there. Oh, wait, no, it's clean. It was just like smear. But yours is dirty.
Because you're quite sensitive, and that's why
Very different. That's why we get on the line first.
only one. Yeah, because it's technically some people actually 4 years, which is why but actually technically hopefully it should be okay.
I just brought
Before you do
they're always like pick up the water off the spot glass. So we're just as a
any other questions?
my brain's like I have to do.
If you guys want to have a look, you can. One very see now we have to so perfect. Oh, that's a yeah, it's still there. It's actually so the issue that I'm going to use that
Or is it just like
Yeah.
that means most of the smaller can
No, but he was saying, I should like learn more. Because obviously they have MPV, they have like a flat or whatever.
I don't have some small class size.
Yeah. I was thinking, haven't done it, they were, like, not pretty hard, more just
Yeah, that's the thing, right? Like, if you have all those extra labs that be created.
What? Lab time? Ew, what's what?
Yo, you think you'll let us go already?
This one's the thing, very.
Oh, one take. I like doing physiology. Yeah,
It's like,
I mean, you could. Then there's like stuff. But then I guess reproductive system. Female, male.
on the third floor.
You need to know like every chemical you use.
Yeah,
You see, it's like oh, okay.
DNI.
It's a part of it.
It depends on other people's like
Yeah. Well, I just like to.
Yeah, they don't want they don't find any.
Like it now.
Like 24 ready to do.
Tomorrow is my happen. He didn't do it. Oh, it's just like.
Understand how like the whole canyon makes the flow race and the convert and stuff like that, and the vascular canyon makes the different stuff like that. You get like probably 10% of that size from just like
There's a lot of like, there will be like a little diagram.
I don't know what. And they can go.
It's little better.
Good idea.
they can take a time to spread it around. It's a very thin layer of
If we had water, it'd still be like. Enough to cover.
Yes, but
This
when
is
you're
mine. I
recording
think I put a tiny bit.
temperature, verify that. I didn't do it.
The beaker. Yeah.
I did.
So like, okay.
That won't, you'll have to add more, I know that.
Yeah.
There is definitely some in there.
So I think,
It's dead.
let's see if,
I think.
like, do the same.
24.2. Oh, I see. No, I see it. Yeah, at the bottom there, it's also a little bit. Yeah.
Yo, are you like trying to put the whole place? Well, I'm. You don't put that much.
Oh, with Daphnia?
Yeah.
With Daphnia, they're already hyper involved.
Yeah.
I'm gonna start.
It's kind of hard to see the flow this slow. Yeah, very. Yeah. I. Yeah. So we'll have a timer for that 10 seconds. And then for that 9 times.
Were you the last two?
Do you want to measure? You'll do the clicks? What? Yeah. The second.
Exactly.
Maybe stop.
Oh. I would say try to swipe out that was the everything around that. You can use both fingers. You can use both fingers.
Yeah. Yeah.
The first step we're gonna do is.
That's how you do it. Yeah. They're starting the timer.
Filters away the water. Filter away so that way it's that should be. Oh, I think it's
It's one way, at least. Water.
Yeah. Okay. Oh.
Then start adding drops of water.
Okay.
How's everyone doing? Everything okay?
Oh, please, yeah.
Everyone else?
So I'm going to talk about the observations, but before I do so, I just wanted to show you another video that will help maybe for those that are having difficulty viewing the heartbeat. This is an example to be able to show you. So the heart of a Daphnia is located in the back near its neck, what you would assume is its neck. This is a zoomed in photo. So use the contrast disc, that black disc, to be able to see more clearly the heartbeat. And that will help you see the Daphnia appear clear. And if anyone is wondering which disc I'm talking about, they're these.
should have a black one. Everyone, I hope you're paying attention. Essentially, these are contrast discs that will help you record or actually compare the contrast of Daphnia. And hopefully you guys got to see the video. Essentially, you have the Daphnia that are transparent, and this helps for you to be able to see more clearly the heart, which is beating right in the middle, shown there. So if you guys have questions on the next position, you could imagine. That long tube is actually their guts. So just bear that in mind. For a few examples, how's everyone doing? Yes. Just for context, everyone has been able to see the Daphnia, yes or no? Has everyone been able to see the hearts, yes or no? You have a problem? Okay. And how many other groups are having difficulty seeing the heart? Yeah. Okay. Okay. So let me just go to this group, down to you, and then to you.
Yeah. I have a water ready in here. Yeah.
When you add the contrast, you may have to adjust the light settings. Bear that in mind. There's also a way to move up. With the first knob, a white knob at the top of your compound light microscope, you move in and out. If you move towards you, it moves down. If you move away from you, it moves up. And then the black knob is helping to encircle the good for the heart for the heart rate.
So we have to talk about that.
So let me clarify. Based on your manual, and have a read of it because it's important to know you're doing every 10 seconds. So essentially, I'm gonna just write it down. You oh, okay, no. So I try to get a visual of the heart. Let's say it looks like this. At the back, that's the mid gut. So you are counting the number of beats that you see every 10 seconds. In the lab manual, so everyone, I hope you're paying attention. Group 4, please pay attention. I need you guys to all pay attention regarding this for actually counting, because it's important to understand that's for recording your data. So using the counters shown there, and you may already have them on your benches, you're counting every 10 seconds. So have a member of your group essentially have a timer for 10 seconds, because essentially the other person is going to be counting with the compound microscope, viewing the heartbeat every 10 seconds, or actually counting up to 10 seconds, how many heartbeats they see. So the palpation, or essentially the up and down movement, is what you're recording as one beat, and you're doing that for 10 seconds. Times 9 because you're doing it for 90 seconds in total. So you can take 5-second breaks. Your manual says 10-second breaks, but if you're well versed, you could let's say start off with like 5-second breaks. But just remember, you're doing this for 10 seconds, 9 times in total. That's for each temperature. Room temperature, you're going to be recording with the thermostat, right? So you need to make sure that you actually have it placed into your large beaker, because that's the one that you're going to be using to record the temperature. And as you use the smaller beaker to add in ice, right, or hot water from 55 degrees, you need to make sure that you slowly thereafter add them into the large beaker, so that way you can simply use your pipette to essentially take out small amounts to place it directly onto the Daphnia. So I'm going to just draw this out on the left-hand side, because essentially the depiction of the Daphnia is just an example here with the antennae and other physiological features. You'll essentially add in 1 to 2, or even maybe 3. We'll say 2 to 3 drops of your final controlled water, controlled in the sense that the temperature is adjusted finally. So remember, you're going from room temperature, going down with ice to 10 degrees. Use your thermometer to keep note of it, ideally around 10 degrees. Then using the 55 degrees water from the water bath, you're going up by 5 degrees. From 15 to 20 to 25 to 30, all the way down up to 35 degrees. And so you're repeating those 90-second, pretty much recording 10 seconds each time for all 7 temperatures. When you're done that, and I know that sounds maybe overwhelming, but you guys can take turns, right? And beautifully, if you think about it, because it's only 1.5 minutes per temperature, even if it takes 2 minutes to account for everything, adjusting for temperature as well, you can literally be done this entire experiment in under half an hour. So just bear that in mind. Accumulating all that data, your other group members can help support, like recording the data itself, the raw data, while you keep count of the time and or the number of heartbeats that you see. But essentially, once you're done all of those calculations of heartbeat, I'll put HR as just like an intonation. But once you're done that, you'll calculate the mean, and then you'll actually, we'll talk about what's next. Any other questions regarding that at least? You'll essentially calculate the mean for all of these different values. There should be 7 including at room temperature. So just bear that in mind. 1, 2, 3, 4, 5, 6, 7. Perfect. Any questions? Everyone should hopefully be able to see now the heartbeat of their Daphnia. Okay. Oh. Yeah. If anyone else is in line,
Yeah, I don't know. Yeah, every time. Yeah.
That's actually. You guys can get read it. That can be done. I will recommend.
yeah.
Yeah. Can we speak? One is that. I worry that you may or at least the ability of cash
If your group Daphnia passes away, they have just let me know actually. Okay. Are we supposed to?
It's the average. Yeah.
If you guys have any questions, let me know. I'm gonna also be on the other side because I realize
Looks like he's sort of, like inside of his bend is on this side.
let's see how it you can also let me know.
Oh, we'll see. Sounds good.
Your group will be
This is your root, most of the root. Yeah. Yeah. Go and say, Yeah. $15. 14.3 if you want to be that. Should be occurring. No, it's not. That's it. Something one number above the low here. That is 6.5. Bro, why? I don't know. Anybody? Yeah, I got. That was a lot.
Yeah.
Yeah.
I added the temperature and stuff.
Yeah. Oh. You can put 50, but you want exact. Like I said.
I know this group has a question. I'll connect you guys.
They're probably doing that. This is the way the things we're looking at the microtension. Really, you could have seen. It is going to a lot of this. It's funny because we're solving a lot of. But I would also say, no, there were it's an issue. Yes. Not looking at the star. Yeah. But just bear in mind that if it doesn't, then
Yeah, I think it should be good. I think it will.
Oh, this.
keep it up, keep it up. Next time. People should.
Alright, let's do water.
So I had a question from that side.
Oh.
Yeah. Okay, now.
3, 2, 1. Three, 2, 1. Higher than finish.
The average should be like that. So, because of swamp temperature of the water, they will.
Three, 2, 1.
You know what, right?
Yeah. Taking your water.
Also, just as an update, if you guys have been working in more than 3 in your groups, that's okay. In fact, we use less Daphnia in the class, which is better. But just bear in mind, you're not adding in the water directly to the Daphnia. you actually are controlling it to start off first with your large beaker, as I showed, or sorry, first your small beaker, then your large beaker to adjust the temperature.
Is that 19? Yeah, I was just.
So some of you may also be a low heartbeat, or like a heart rate that's quite low. Generally speaking, you should get them in around 20 for many of you. If you aren't, then it's likely the case that you haven't added enough water and it perhaps is in the midst of actually failing in life. So just bear in mind adding more water. Make sure it's not being dehydrated. Remember, this is a water thing. So you need to have enough water. Otherwise, if you're given very limited amount of water, then they run the risk of survival. So give it, if you want to drink over 3 drops, that's okay. 5 drops is okay. But just make sure that you're regularly changing the water. So once you go to a new temperature, be it from room to 10 degrees, you remove the room temperature water, even just 3/4 of it, so that way it's still suspended with the water, and then you add in the ice from your large beaker. Just bear in mind that you don't dehydrate the water, please. But generally speaking, if you have like a heart rate that, actually let me know your heart rate, and then I can let you know how they work, how they. What was it?
30.0. This is not 35. That is right.
Towards the center of your head. Do not leave things towards the edges because sometimes groups have actually spilled and broken glass. So please be mindful that you're not things that are away from the edges, including the thermometer. Follow the protocol of methods in the law manual. It's okay if your observations are unique, but it doesn't mean that you want to just forsake what you see in the method. Follow through what you see. When it comes to flushing out new temperatures of water, you take it out first from your petri dish with the Daphnia, and then essentially add in, take out at least 3/4 of what you see in the dish, and then add in more water accordingly. A few drops, three drops should be okay for the new temperature that
Yeah. Like our answer very like linear, like it goes up by like 4 dishes every time. I think it's good. Yeah. Yeah, I think it might be like time of day. Yeah, I thought we should do that actually.
Just like double check that's an okay result.
Yeah.
Oh, yeah. Oh, yeah, done. What's the They're going to take I want to know the feeling
Yeah. Go back. Start off.
We have a question, like, when you're not started, or like,
Snowball. That's good though.
really?
It should be between like 1 and 6 inches.
Oh, okay, okay.
Also, when you're a sheep, like for a cat that is very slow,
Yeah.
when we hit him with the cold water, it's
Does look crazy? Oh.
just like, it's like a vibration.
Oh, like,
If I have to take another bath.
but, oh, I think ours was great. Not that ours was great, but- I mean, it's funny. You do. It's actually, I just put that
I'm glad I never.
We're measuring that.
Oh, that's right.
You guys, if you have like amazing observations, record them. It's gonna be really exciting for yourself. And you can think about it like down the road.
Okay, alright.
If you find sense, what is the treated plant? I mean, because I mean, I don't. The range for your heart rate for Daphnia should be 20 to 60 beats per 10 seconds. So if you're seeing things that are obscurely lower or higher, you should let me know and we can help hopefully resolve it. You may otherwise need a new set of Daphnia.
Like between 30 and. Can you please prepare?
Let's see. Because technically there's 4 of you, so technically there's
Then I got it high twice. But that was pretty much perfect. A little bit of yeah, it's been going on.
That's great.
You know what? Yeah. That's what it's like. Why did I share it job? That's what What doesn't like usually it's because it looks like it's like you really like the music is that's it makes you
I can't really hear you.
Yeah, rather than. You guys can think if you can.
Yeah.
Yes.
I
forgot about.
Yes.
Oh, is it just industry? Can I have a look?
So it's still pretty fun. Yeah.
Just before I answer any questions, how's everyone doing in terms of timing? I'm just gonna see the time here. Okay. So we have about an hour, under an hour left, because we want to finish by 12:40. Even if like you're stuck, I'll give you 1 hour or under an hour at least to complete today's practical. But I want to get an idea for the number of temperatures that have been completed thus far. This group you're at 50, going to 20, right? What group what temperature are you guys on? Okay, so three to four left. How many more from your side? Three. How many more from your side? Four. How many more from your side? Three. From your side, 3 to 4 left, or one? Okay, one more. One. Four left. Oh, okay, no problem. So here's what we're gonna do. Continue on as you are. At about 12 o'clock, I'm gonna go over data analysis to try to finish as much as you can, because essentially, remember, each recording should be 10 seconds for the number of heartbeats, meaning that for one temperature, because you're doing it 9 times for 9 values, you should be done under 2 minutes. And then like add in new water. Someone from your bench could do this at the meantime. And then essentially, like even if it's 3 minutes for one single temperature, you should be done in the next 15 minutes. That gives you an extra 5 minutes in case anything urgent comes up and you need my help. But just let me know. Hopefully about 15 minutes, 12. If you can be done before 12 o'clock, that'd be even better. Thanks a lot.
No, this is not better than you got. Maybe so. You're all things about. Yeah.
I have to keep that.
Yeah. Yeah. So we got about hours. You did between that time,
How does
or if it goes up,
Back here, but one. Cause I have a lot of it. Right.
Okay.
If we're done by the. Yeah, it's if we're done by 12.
Did you look at the longer view?
Yeah, that's pretty close.
Okay, so just want to confirm from everyone else. For the groups on my right side, the left side of the practical room, how many more temperatures are left? 2, 3, 3. Like 3 whole temperatures? Okay, let me have a look. Reason is actually if you may have so it's not like we have these. So don't say
I can.
In 5 minutes, I'll be likely going over the data analysis that I've done. They could begin looking at what next steps they need to do to complete today's practical. Bear in mind that with the Daphnia, when you're cleaning up, you're placing it inside the sink. Do not just place them into the garbage. They are, after all, organisms, and they are water free. So I'll help you rinse them off once required. Then once you're done, you can place back all other belongings, shut off the microscope, dry off the plate as well, into the BBL.
I don't want to take away your thing. Yeah.
Yeah.
Yeah. Yeah. Yeah. That's a G. That's all. Yeah. It's shows that there's years. And there's I could all. So it's not like his answers are. Yeah. Yeah.
Yo, you guys don't even have that. Yeah, but it's easier if you want.
Yeah, because it would be there. And oh yeah, yeah, yeah. In your photo, don't listen to these calculations. Like listen to the answers, but not. Yeah. Yeah.
It's on the left now, turn it around. Part is on the left. Did you see it? It's the glow. So, what do you think it is?
Yeah. Yeah. That's fun. Because I wish they girls like bioanalytic I usually just look at the last yeah, exactly. Yeah. That's exactly like this. Like literally, if you like understood like every quick action, yeah. That's most effective. Most professors are private close. Yeah. 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 We get that. We get
Before actually I answer further questions, I wanted to also bear in mind the timing, right? Because we do have, as I mentioned earlier, we're going to be talking about data analysis, right? Apologies for the timing, because I have to as well keep in mind those that had other questions. But let me just briefly end off or summarize the rest of today's practical. So the filtering tube, that's separate and perhaps hopefully answers the question for group 5. One of the groups had apparently Daphnia that is pregnant, so they're able to see beautifully an enlarged Daphnia water flea. If you want to have a look, it is the middle group on the right-hand side. As well, pretty much just to summarize, we already talked about most of this, but to repeat for those that are wondering, you need to be able to collect all of this data for your lab assignment. So that includes all of the 9 values or heart rates per 10 seconds of each temperature. And so recording that for all 7 temperatures, you should have not only that data, but the mean values for heart rates. And that would be measured in beats per 10 seconds. Bear that in mind for your table. Bear that in mind. Hopefully everyone's paying attention. Everyone paying attention for your, not only for your table, but for your figure caption, your table caption, your graph. All of them need the appropriate units, and you need to provide specific detailed information. The organism, the temperatures, the variables that you're accounting for, and their units. And that's something that I want you to showcase throughout your lab assignment. So that way you can get full marks and also help prep yourself for the FLR. Once again, just be patient as well when you're changing temperatures if you're almost done. If you also have questions or concerns, let me know. I mentioned this earlier, but 20 to 60, even in our case here, 20 to 40 heartbeats should suffice. But if you have them lower, then you can describe why in your lab assignment or higher, respectively, if it's over 60 heartbeats per 10 seconds. Now, I just want to briefly go for the exercise. Folks, I hope you're paying attention because it's very important. You have 2 formulas to deduce. Okay. Sorry. So you have essentially for exercise 3, the data analysis of your heart rates. You measure them for every 10 seconds. You calculated the mean across temperatures. And now you're getting your Q10. Your Q10 is a correlational statistic to derive how sensitive a parameter is to temperature, in our case, heart rate, right? So what you need to keep in mind is that you have 2 formulas to deduce this. If it's over 2, that signifies it's very sensitive, right? Heartbeat would be, or heart rate would be very sensitive to changes in temperature. And if it's below 2 and closer to 1 or even below 1, then that assumes that there is a low correlation between that biological rate, like the case of heart rate, over temperature. Now you have 2 formulas to deduce this. Formula 1 is for essentially any range of temperatures, whereas Formula 2 is for exactly 10 degrees centigrade apart. Formula 1 uses logarithms, so be familiar of how you would use them with your calculators. You have also sample calculators here that you could use for the rest of today's practical. But the label is also shown for the different variables and what they signify in your manual and also here on the slides. Log R higher signifies the rate of reaction or the heart rate at the higher temperature. That's for your mean values. Keep that in mind for all formulas, that the R represents your mean heart rate. So that would be RH for the formulas. And then you have essentially for the log one in the numerator, both heart rates for the higher and lower temperatures. And then in the denominator, you have both temperatures similar to essentially x2 minus x1 like in slopes, which is actually relatable for microbiology because this is actually something that I'm taking as well to determine generation time in a similar way. Now Q10, if your formula for number 2 is exactly 10 degrees apart, like 20 degrees to 30 degrees, then what you're doing is calculating your Q10 for the higher rate or your heart rate at, for example, 30 degrees divided by your heart rate at 20 degrees. That is your Q10. It is that ratio. And essentially, for the Lab 3 assignment, you're including all those details that I mentioned before. You're including all of these raw values, right? So that's, let's say, part A. Part B is the mean values. Remember, you keep in mind the units and the decimal points. You can go up to 2 decimal points for this lab, but some labs are more specific, and even in the assignment, it makes mention to go up to 1 numerical digit for another question, 2 other questions. But the point is, this is part A. All of your raw data, including room temperature. If you have other groups' results, you can include them, like for the cases of the 2 individuals on that side, or like from this, on this side of the room. Overall, you have part A, part B, the mean values. Part C is actually your graph. So the graph is going to be essentially the temperature. You want to provide more specific details, but for simplicity, I'm showing that you're having temperature in degrees centigrade or degrees Celsius, and over degrees centigrade, you have your heart rate, which you're going to specify the units, right? It's the number of beats every 10 seconds, not every minute, but every 10 seconds. Bear that in mind when you're including those values. If you want to adjust the units, you can, but just bear in mind that you're doing this correctly. It says as well to include a line graph. I want you to please place close attention to this because the increments go up by 5, right? You start off at initial, which could be 24 degrees or another increment. But essentially, apart from that one, everything else should be aligned, all going in order from 10 all the way to 35 degrees, and you're having your room temperature one somewhere in the middle between 20 and 25 degrees. All of your data points, the mean values go up, or essentially you record them as you expect them to be seen, and then you can use a line to essentially draw out the curve. You can also use other statistical analysis to draw a line of best fit, for example. Apart from that, remember that even for your text box or your caption, you keep as much detail as possible, right? I'm keeping an eye for those that are also using their phones. Please be mindful that you don't want to get any marks deducted, so just focus, pay attention, because these are details you don't need to get deducted for, right? So bear that in mind, be as descriptive as possible. It even says 2 to 3 sentences. For your FLR, you may even need to be more specific, perhaps 5 sentences long. So bear that in mind, especially for details, for your figures. Make mention of what you're aiming to uncover or aiming to explore or find, and be very specific as to the conditions you're using, the organism, the variables that you're accounting for, the time points, the temperatures, etc. So just to bear those details in mind, because that will help you down the road with future assignments and future courses. So I finished a minute over, technically 12:15 was the aim, but nonetheless, alhamdulillah, we're almost done. Reminder for everyone, clean up. So actually, before I say clean up, everyone done? Everyone done? All values, or at least all raw values. Hopefully you're able to calculate the mean as well. So that way you can complete the Q10s. You can also stay along here if you want to complete the rest of the practical, or actually better yet, do them when you can. There is this other question about your plates, your Daphnia. Please, once again, do not dump the Daphnia into the garbage. Place them, so if you have your plates and you're done, shut off your microscopes, have one person do that. Bring it, let's say that's your plate, you have Daphnia inside. So this is what you do. Your beakers, if your beakers have water, you're just pouring on one edge the Daphnia, so it's just briefly and slowly essentially flows off and down into the sink, down into the drain. This is my more ethical approach rather than just like killing it, because after all, these are organisms like ourselves. So we have to be mindful, right? As studious scholars, polymaths, prodigies, or scientists, you want to bear in mind that you're following the most appropriate standard. So bear that in mind. You see ice around the location of where you want to put in water, just rinse off the water or take off the ice, and you can add in some of the aged water here. Don't add in too much because you should be able to remove this with gloves or with paper towels. Take out the ice so that way you can safely place it in the Daphnia without it having to clean it off. You just bear that in mind. Apart from that, you can clean up prevention, please back to counters, sample calculators, pens, anything that you have. Shut off the microscope and have an amazing display before seeing it. In the zoo. Take care, everyone. If you have any questions or problems, let me know. Don't forget your FLR. You on March 3rd. Okay, so by the way, everyone needs to clean up your benches, pantry dishes, need to be cleaned up with chipmunks or as well. What are you concerned? If you have any data, oh, yours, okay. Oh, that's it. Is it okay? Is it okay? Yeah. You guys can. If anyone wants to have a look, okay, don't like obscure the delivery of the newborn Daphnia. But essentially, all of the groups make sure that you clean up appropriately before you leave, because that's part of your responsibility. And then accordingly, just make sure you dry off your bench top with the dishes as well. With that, have an amazing reading week. If you have any questions, let me know. Looking forward to your excellency in this life and the next. Take care, everyone. Have an amazing reading week and all the best in your endeavor. Take care. I want to look at it again. If you guys want to. Actually, you can go out. I may catch. I don't want to put a time on you. I just go see it amongst the first people there, and I'll be with you. Take care. No. Okay. Anyone having problems on this side? I'm gonna join you guys. Any concerns? That's amazing. Anyone having problems on this? Just like the normal. Just let me know. Keep it up. No pressure. By the way, you're recording like 30 minutes. Don't worry. You can slowly if you want to, like very timidly clean up. And if you have questions, for SOR or anything. Keep it up, you guys. Just a kind reminder, we'll be meeting in Toronto. So please, I don't want you to forget. Remember Toronto 3 weeks from now, 9:30 a.m. if possible. Or even 10 a.m. It takes a lot.
Yeah. Yeah.
Yeah, I've never seen that before.
One else wanted to see the pregnant Daphnia, the hands. But if you have to go, it's that's okay.
Most of them are public.
Sound of pregnant.
Yeah.
The cost of to get it if you want. How is it?
The shoulder. It's about
this. Yeah,
you guys, sorry, with a lot of.
Yeah.
It's a beautiful observation. Okay, take care. That's it. Remember, FOR, strong thinking, all of it. Okay. Thank you.
Any questions, concerns, problems, technicians? All good? Everything okay? Sorry? Oh, the count. Okay. Let me see.
Well, there's one that's broken, right? So.
Okay, so
I'm trying to fix it, that's why.
anything else?
No problem. Yeah, this is I actually really enjoy this exercise. I think especially the pregnant one, they're yeah.